Today we're going to talk about early embryonic development. Let's begin with the chemical process of the sperm entering the egg. The jelly coat that surrounds the egg continually releases molecules that attract the sperm. Once the sperm binds to the jelly coat, it activates a signal transduction pathway that causes calcium ions to be released from the egg's endoplasmic reticulum into the cytosol. Sperm binding also triggers exocytosis of a vacuole in the tip of the sperm head, containing enzymes called an acrosome. These enzymes, released from the acrosome, make a hole in the jelly coat through which the sperm enters. Actin filaments then grow out of the sperm head and penetrate the jelly coat. This is the acrosomal process. Proteins on the surface of the acrosomal process then bind to receptors on the egg's plasma membrane. This lock-and-key gametic isolation ensures that the egg is being fertilized by a sperm of the same species. Next, the sperm and egg membranes fuse, which triggers depolarization of the egg membrane. This prevents polyspermy, blocking any other sperm from fusing with the egg. As we mentioned before, calcium ions are released when the sperm binds to the jelly coat. After the depolarization of the membrane, the cortical reaction occurs. The elevated calcium levels cause vesicles filled with many different molecules called cortical granules to fuse with the plasma membrane. The molecules contained in the vesicles are released into the space between the plasma membrane and the vitelline layer, which is called the previtelline space. These molecules cut off the sperm binding receptors in the egg's membrane, and a fertilization envelope forms. This further prevents polyspermy. Finally, the sperm nucleus is released into the egg. Once this has occurred, egg activation happens, and the rates of cellular respiration and protein synthesis by the egg are generally increased. About 20 minutes after the sperm enters the egg, the sperm nucleus merges with the egg nucleus, creating a diploid nucleus of the zygote. Then, DNA synthesis begins and the first cell divisions occur.
After the cell is fertilized, the zygote begins the first series of cell divisions called cleavage. This stage involves rapid mitotic divisions without cell growth. These divisions split the zygote cytoplasm into smaller cells that are called blastomeres. Each blastomere has its own nucleus. The blastomeres continue to be cleaved and become a morula. Inside the morula, a fluid-filled cavity forms, which is called the blastocoil. When the blastocoil forms within the morula, this produces a blastula. So, in summary, after the zygote is fertilized, cleavage forms blastomeres. Then, the morula and the blastocoil make up the blastula.
The next stage of embryo development is gastrulation. During this stage, the blastula undergoes a drastic rearrangement. This rearrangement of the cells results in the formation of three distinct layers in the embryo. The innermost layer is the endoderm. The endoderm layer forms epithelial linings of the digestive, respiratory, and excretory tracts, the pancreas and the liver. The middle layer is the mesoderm. This layer forms the skeletal, muscular, excretory, circulatory, reproductive systems. The mesoderm layer also forms in animals' blood, bone, and lymph. The outside layer is the ectoderm. The ectoderm forms into the animal's skin, nails, teeth, lens of eye, and the nervous system.
We are going to explain the motion of gastrulation using a sock model.
Gastrulation begins with an indented crease in the blastula. This is formed by cells pushing inward. It is called the blastospore and will eventually be the mouth or the anus. Sheets of outer cells roll inward, forming the mesoderm and the endoderm. Cells at the opposite end of the embryo stretch over the outside, becoming the ectoderm. Next, the embryo goes through organogenesis. This stage varies significantly between different types of organisms. In all organisms, the three germ layers will develop into specific organs. During organogenesis, cells migrate, condense, signal between tissues, and change shape to generate new organs. Organogenesis is very similar between vertebrates. Here's the process in more detail for a frog embryo. One of the first organs to form is the notochord, which is a skeletal rod. It is formed from the mesoderm. The ectoderm above the notochord becomes the neural plate in response to cell signaling. The plate rolls inward, giving rise to the neural tube and the neural crest. The neural tube will become the center nervous system, which is the brain and spinal cord. Some of the cells in the neural crest migrate to other parts of the embryo and become a wide range of things. Next, somites are found from the mesoderm, or formed from the mesoderm next to the notochord. They are blocks of cells that become arranged around the notochord and become vertebrae. Portions of the notochord between the somite become vertebral discs. Some somites migrate away from the notochord and become muscle tissues around the spine and ribs. The mesoderm next to the somites become lining for the body cavities. After these initial stages, cell differentiation continues and organs are refined.
In conclusion, the 4 stages of early embryonic development are fertilization, cleavage, gastrulation, and organogenesis.